In a recent video, Dr. Jared Younger, head of the Neuroinflammation, Pain and Fatigue Lab at UAB, provided an insightful explanation on the various triggers of chronic brain inflammation and microglia priming. Dr. Younger highlighted pathogens, environmental exposures, medications, and internal bodily states as key factors. However, despite the comprehensive overview, there's a significant omission in the discussion: the role of electromagnetic fields, or EMFs. This blog aims to bridge that gap by highlighting the overlooked impact of EMFs on microglia and the broader implications for public health. Microglia are the primary immune cells of the central nervous system, acting as guardians that protect the brain from pathogens and other threats. When activated, they produce pro-inflammatory cytokines to combat these threats. However, chronic activation or priming of microglia can lead to persistent neuroinflammation, contributing to various chronic conditions such as chronic fatigue syndrome, fibromyalgia, and neurodegenerative diseases like Alzheimer's and Parkinson's. Dr. Younger discusses three main conditions that can lead to microglia priming. Severe infections. Life-threatening infections that invade the CNS can cause microglia to remain in a heightened state of readiness, producing continuous inflammation. Chronic low-level exposure. Continuous exposure to low-level immune insults, such as toxic substances in the environment, can sensitize microglia over time. Repeated acute exposures. Frequent, closely timed exposures to immune triggers can overwhelm microglia, leading to their chronic activation. While Dr. Younger's discussion is thorough, it misses a critical environmental factor: EMFs from wireless devices. There is a significant body of research indicating that EMF exposure can also prime microglia, leading to chronic neuroinflammation. Studies have shown that EMFs can act as a continuous low-level immune insult, similar to the toxic exposures mentioned by Dr. Younger. EMF exposure has been linked to several critical issues. Firstly, oxidative stress. EMFs can increase oxidative stress in the brain, a key factor in neuroinflammation. The study by Zielinski et al. In 2020 demonstrated that EMF exposure can induce oxidative stress and autophagy in neuroblastoma and microglial cells, highlighting the potential for EMFs to act as a chronic irritant to microglia. Secondly, pro-inflammatory cytokine production. EMF exposure can lead to the production of pro-inflammatory cytokines by microglia, as seen in various studies on the effects of RF radiation on brain cells. Lastly, disruption of cellular functions. EMFs have been shown to alter the function of microglial cells, impairing their ability to perform critical tasks such as phagocytosis. The study on curcumin's ameliorative effects demonstrated that EMF-induced pro-inflammatory responses in microglia could be mitigated by curcumin, suggesting that EMFs indeed play a role in activating these immune cells. Connecting EMF exposure to neurodegenerative diseases. The research by Steffi et al. In 2019 found that EMF exposure alters the metabolism of amyloid precursor protein, or APP, and alpha-synuclein, proteins implicated in Alzheimer's and Parkinson's diseases. This suggests that EMFs could contribute to the pathogenesis of these neurodegenerative diseases through chronic microglial activation and subsequent neuroinflammation. Evidence from major studies. Several major studies and reports have highlighted the potential health risks associated with EMF exposure, including the Interphone study, which found increased risks of brain tumors associated with heavy mobile phone use. The Hardell Group studies showed a correlation between long-term mobile phone use and brain cancer. The U.S. National Toxicology Program, or NTP, demonstrated clear evidence of carcinogenic activity in rats exposed to RF radiation, with significant findings related to glial cells. The Ramazzini Institute study confirmed the NTP's findings on the carcinogenic effects of RF radiation. Additionally, the Bioinitiative report and research by Dr. Henry Lai provided comprehensive reviews of the non-thermal effects of RF radiation on biological systems. Conclusion: bridging the knowledge gap. Dr. Younger's video provides valuable insights into the triggers of chronic brain inflammation, but it falls short by not addressing the role of EMFs. As the body of research grows, it becomes increasingly clear that EMFs from wireless devices are a significant environmental factor that can prime microglia and contribute to chronic neuroinflammation and neurodegenerative diseases. It is essential for healthcare professionals and scientists to consider EMFs in their discussions of pro-inflammatory exposures to better inform the public and guide protective measures. Call to action. We urge researchers, clinicians, and public health officials to acknowledge and address the potential health risks posed by EMFs. By integrating this knowledge into broader discussions about chronic inflammation and neurodegenerative diseases, we can take more comprehensive steps to protect public health in our increasingly wireless world.